So chapter 6, again, is about metabolism, about energy, and looking at how that relates to life. And also, energy is what allows life to continue. Okay, the moment you reach what's called equilibrium, or where there's no energy going out, no energy coming in, you what? You die. And so basically that's what death is reaching that state of equilibrium. Okay, here in a little bit we're going to look at what's called entropy. And entropy is basically the state of disorder. Okay? And so one of the properties of life is actually life is order, has order to it, has specific qualities and traits of it. And as you go towards that state of entropy, you actually become less and less disordered, but life continues to try to make itself more ordered. So it's that constant balance. So I want to show you a little bad video about energy and cells. We're not going to watch the entire thing.
I know what I'll do. I have the 3-hour energy for just $29.95. I get 3 hours of sleep. Who needs sleep with 3 hours energy? Tastes like trash. Hang on. I know what energy is. I know that chemical reactions create energy. Well, what does myself do with the energy? Alright, I'm done with the terrible acting. What's going on, everybody? My name is Jack Nichols, and this is Academy of One. Today we are talking about energy in the cell. I got my 3-hour energy, and I am pumped. Let's go into detail about how cells get their energy. Plant cells get their energy from photosynthesis, which is its own video. So we'll talk about animal cells. Animal cells get energy from the breakdown of glucose. The glucose will combine with oxygen and release carbon dioxide and water. This breakdown is how cells get energy. The whole process will be explained in more detail in video 5.12, but this is good enough. So now we have energy from the glucose, but it can't be used. At least, not yet.
So, not saying it's a bad video, but Jack did a good job. Thank you, Jack. Come on. There you go. So a couple terms. Again, these are going to be on your vocabulary list that you do need to know, and I'll kind of use them interchangeably. Two different ideas, what's called catabolism and anabolism. And catabolism is where you are releasing energy to break it down. Okay, so again, that whole, I refer back to the dude you're playing like Betty White commercial, the Snickers commercial. The reason why he's playing like Betty White is because he doesn't have enough energy. He eats a Snickers, he's going to break down the energy through the catabolism to release energy. Whereas the opposite of that is anabolic. Anabolic is where you're actually building these energies. So you can actually look at the two processes, things like respiration and photosynthesis. Whereas on catabolism, which one is that? Is respiration or photosynthesis releasing energy? It's respiration, right? And whereas anabolism or anabolic is using energy, so that would be photosynthesis. So you're using the light energy to create, and then you're using catabolism to break that down in the process of respiration, cellular respiration. So again, we talked about kinetic and potential energy, the cute little girl on the slide. Remember, this is stored energy due to position, but in biology, we're not so much worried about the stored because of gravitational. What kind of energy were we looking at? Potential in the form of, the release of energy through heat and through the breakdown of these bonds, these chemical bonds. So this chemical potential energy. So most of the time, we're not so much looking at the little girl going on the slide, we're more looking at the carbon bonds, and you're eating lots of sugar. You're eating lots of donuts, and that has lots of chemical potential energy. So there are actually 4 laws of thermodynamics. You need to know the first and the second law. There's actually a 0th law. So it could actually go 0, 1, 2, and 3, but there's 4 laws. Weird, right? So the first law basically just is the law of thermodynamics, which says that all energy cannot be created nor destroyed. Right? You've heard that with mass, you've heard that with matter, and all those different things, right? So the first law just says that energy cannot be created or destroyed. So whatever energy you use, you're not really destroying, even though you're using it, you're just changing it into a different form. Okay? You're changing it, most of the time we're changing it to heat. Okay? The energy that the sun sends to us, the energy is not being lost from the sun, it's just being changed into chemical energy through photosynthesis. And the second one is the law of entropy. Okay? Your room has the standard law of entropy. That's the law of disorder. So how's your room like the law of entropy? It's messy, right? You go and you got to go clean your room, and you got to make sure everything's very stable and everything's good. And then like a week later, like, oh, really? Because I just fixed this. Okay, that's entropy, and it's a measurement of how disorder something is, and the universe itself is actually going towards a greater state of entropy. Okay, whereas biology or the life actually wants to maintain that order, so it actually is fighting against entropy. So your body is naturally decaying. Okay, your body. The reason why cancer occurs is because your body is actually releasing these free radicals, and those free radicals is actually what's causing cancer. It's causing an overgrowth of cells, and so as your body is going towards a state of disorder, your body is trying to fight against that entropy, and it's trying to create more order. So it's going against the laws of thermodynamics.
So entropy is attempting to fix disorder, or
Entropy is disorder. Entropy. The more entropic something is, or the more entropy it has, is the more disorder, the more messiness. Okay. The next thing you need to look at is what's called free energy. So on free energy, you're going to a state of equilibrium. You're going to a state where the energy released is equal to the energy supplied, and that's what's equilibrium. That's where you get to the point of death on life. Okay? If there's no energy coming in, there's no energy going out, that's death. So there's two different types. There's what's called exergonic and endergonic. Okay? When energy is being supplied, where the reactant's energy. Okay? If you measure the amount of energy in a walnut, okay, and then you burn it and you say how much energy is being released. In a walnut, you've got the reactants. You burn that walnut, it releases a lot of energy, so the amount of energy left over in that walnut is lower. So that's what's known as exergonic, whereas endergonic is where it's actually increasing. Okay, this would be things like photosynthesis. The energy in that cell, or the energy in that carbon molecule is very low, but the energy in the product of glucose is very, very high. Okay? So if energy is going in, it's endergonic. If energy is coming out, it's exergonic. So most reactions will be exergonic. Some reactions, things that actually become colder, are endergonic. Okay, because it's actually pulling in energy. There is an equation down here. You don't necessarily need to know how to use this equation, but do know a couple things. First off, what energy is actually available to do work on this equation? Take a look at the what delta G, H, and S are, and then T. You know what enthalpy is? You ever heard that term? In physics or chemistry, enthalpy is basically the measure of energy in the form of heat. So can we use heat to do work? A little bit, right? But is it maintainable? Is it very useful work, energy? No. So most of the time, the delta H is actually useless work. It's useless energy. So what we basically have for free energy is how much entropy, or how much change there is in the disorder. And the more change in disorder, the more free energy, the more available energy to use. So if you think back on ATP, ATP is, if you think back to your previous biology classes, ATP was the energy source. It was the money for any kind of biological materials, any kind of biological organism. So you do need to kind of know that the structure of ATP, it is adenosine triphosphate. That's actually what ATP stands for. So adenosine, the A, and then tri is in 3 phosphates and the phosphate. And you think back to the functional groups, phosphates are there to transfer energy from one organic molecule to the next. So you do have that ATP. Now the process of regeneration is this little small diagram right here. And so essentially what you have is any exergonic reaction. Again, an exergonic reaction is a reaction where energy is being released And if you take that energy being released, it actually creates ATP. Now, to break that ATP apart, you have to use water. So again, it's going to go to a hydrolysis. Hydrolysis is going to break that ATP apart, and it's going to have an endergonic reaction. That's going to be used when it's storing energy. And then that then breaks, it uses it for, it adds itself, so it goes into ATP, adding water, then releases, and it goes to ADP, or as in diphosphate. You then add an extra inorganic phosphate, and it creates ATP again. So it has a cycle here. So that doesn't seem super clear, so let me try one more way. So let's say, for instance, here's the adenosine the adenosine, the adenine and the ribose sugar. And each of these squares represents a phosphate ion. So as you use phosphate ions, there is a bond between each of these. And that bond is stored energy. It is CPE, or chemical potential energy. Unlike gravitational potential energy, this is energy stored directly in these bonds. So every single time we break one of these bonds, here's this one right here, every single time we break this outside bond, what happens is that that bond breaks off. This phosphate will go off, and it will make ADP. That break in the bond releases energy, and the ATP now is ADP. So now what you have is ADP. The ADP is not nearly as powerful, it has nearly as much energy because it has less bonds than ATP. So you have to actually add that on to another phosphate. So what you do is you then add that organic phosphate into a different process. So we're going to go back and we're going to have our ribose and our ADP. You remember ADP is just two phosphates. And so this inorganic phosphate right here will actually join together, making ATP. So when you look back to anabolism and catabolism, this right here is catabolism. We're actually breaking bonds to release energy. Whereas this one, we're creating bonds to form a new product to store that energy. Okay, so this would be like respiration. Respiration is releasing that energy in the form of glucose. Anabolism is actually like when you go through photosynthesis. So and then it stores that up. That ATP then goes back and is reused in the respiration. The respiration breaks off the phosphate. The phosphate releases energy. It creates ADP. The ADP then goes back across into an anabolic reaction. The anabolic reaction rebuilds that ATP from ADP, and it goes back and forth. So it's a continuous cycle. So it regenerates that way. Next idea is enzymes. Now, an enzyme is a type of catalyst. So if you remember a catalyst, go back. A catalyst is something that actually speeds up the reaction of the reaction itself. So you have this catalyst, this enzyme, and this is actually a drawing or a space-fill model of an enzyme. You notice there's basically three parts. There's the enzyme itself. So again, there's the enzyme itself, the active site, and then the substrate. So it's also known as the enzyme substrate complex. This enzyme, and its main idea is to lower the activation energy. So the enzyme lowers the activation energy. So if you've got a graph, and this graph represents basically as you go up the y-axis, it's the energy as it increases. This is the amount of energy needed, and this is time going on the x-axis. So as you draw on this graph, let's say this is an exergonic reaction, you have the reactants. The reactants start right here. So the reactants start right here, and they go up, and they reach a certain point, and then they go back down and release that energy in the products. So you get the reactants and the products. Well, that's your basic energy reaction. And this point right here that highest point is known as activation energy. Well, what enzymes allow you to do is lower that activation energy. So the enzymes themselves start at the same place as the reactants. They go up, but their activation energy is lower. And since their activation energy is lower, then it uses less energy, it uses less time, and so then the enzyme is actually able to work much quicker. Enzymes are substrate specific. It used to be thought as a lock and key. So lock and key is this idea where basically I can't use my keys to get into another room, and they can't use their keys to get into my room. But my key will fit into my lock, and so that's the same idea. This substrate, this sucrose substrate here, can only fit into this lock. Okay, so like I said, that was a previous idea. Well, you had a lock, and you had a key, or you had an enzyme and a substrate, and that substrate locked into the enzyme, and then it went through a hydrolysis reaction and broke apart the two parts and ended up having a product. And notice then again the enzyme is reused. These enzymes do not break down. That's a major idea here. These enzymes do not break down. They are continually being reused. But that's not how the thought is now. The thought is now is that you have what's known as a induced fit model. Okay, the induced fit model basically says, yeah, sure, you've got this lock here and you've got this key here. But if your key doesn't fit exactly right, just like a lock that doesn't fit exactly right, a key doesn't fit exactly right, you can shake that key, you can jiggle that key enough to be able to unlock the lock. Well, it's called the induced fit model. It's another idea like my daughter, she's building with her blocks, and she's got those little that bucket with the different shapes of holes. And she wants to put she's going to put the round peg in the round hole and the square peg in the square hole. But then she sees a triangle, and being the GT student that she is, she's going to think that there's a triangle in this square, so I should be able to fit my triangle into the square hole. And so she's trying, it's not working, so then she gets her big mallet and she starts whacking away at it. Well, what she's doing is she's inducing a fit. She's making her triangle peg fit into a round hole or a square hole. And that's basically the idea, is that basically this substrate here, okay, this substrate can fit directly into this hole, even if it doesn't fit exactly right. So this hole is the active site, the substrate, and so that way the enzyme can start working on it. And again, these enzymes are allowed to lower this activation energy because it helps basically process this idea. And just kind of a little point, just kind of a little reference point. Remember, if you see anything that ends in ACE, it's an enzyme. If you don't know what the name of the enzyme is, take whatever's being broken down, throw an ACE at the end, and so then it becomes an enzyme. So something like lactose is broken down by lactase. Maltose is broken down by maltase. Peptide bonds, which peptide bonds again are what joins two amino acids together to form a polypeptide or a protein, is broken down. The peptide bond is broken down by peptidase. If you're going to break down a couple of toothpicks, it's toothpick ACE. If you're going to break apart some paper, it's paper ACE. So when you're writing your essay and you don't know the enzyme, just throw ACE in there and you might get an extra couple of points. So there are a few environmental effects. The first one is temperature and pH. That's this idea right here. This idea right here. So if you notice, your human body, okay, your human body is at that 98.6 degrees, or on Celsius level is right here. Okay, your enzymes in your body work perfectly at that point. You notice that the rate of reaction is the highest right at that point. It does increase as you're healthy. Okay, so you still have a good reaction rate. But notice what happens when you hit that 100 mark or at like the 45 mark. The activation energy, the rate of reaction, not the activation energy, the rate of reaction decreases significantly to the point of 0 when you get up to the 103, 105. So think about it. You get a fever, you're going to go past that. These enzymes aren't going to work as well. That's why you feel horrible. That's why you don't feel good. That's why your processes stop working. But then on things like hot springs prokaryotics, like the archaeobacteria, the extremophiles, they're going to live a much higher area, and their optimum temperature is much higher as well here. So this is the optimal temperature, and it goes back to this idea, back to like the previous test, of denaturization. And again, if you remember back to your vocabulary terms, a denaturing protein is actually changing the structure of protein due to breaking of the hydrogen bonds, breaking of the disulfide bonds, breaking of any kind of covalent or ionic bond that's been formed between the alpha helix and the beta pleated sheaths, or two other parts of the protein. And so another way you can denature is also using things like pH. So just like how we have an optimal temperature, we also have an optimal pH. So your stomach uses pepsin. Pepsin is an enzyme that breaks down parts of your food when it's digested in the stomach. So your stomach's acid, hydrochloric acid in your stomach, is between the pH range of 2 and 3. But if you take that enzyme and throw it into your blood, which is right here, the pH of 6 to 7, it uses things called buffers to maintain that pH. So you have these buffers, and it keeps your blood between about 6 to 8. The pH of 6 to 8. So if you notice right here, the optimal place for rate of reaction is right here at 6.5, whereas in your stomach it's about 2.5. So again, this enzyme right here will not denature if it's in this low temperature or this low pH. But this enzyme, this trypsin, will denature in that very low pH. So we're going to look at that in an enzyme lab we're going to do later on. Next idea is these cofactors, and there's going to be a video playing. So it does play very quietly because I recorded it from the speakers. But basically what you're going to look at is what's called a cofactor. And a cofactor is like things like vitamins and those ideas, also known as coenzymes. And what they really are not truly enzymes, but they basically help with the enzyme. A lot of these are metallic ions. Those metallic ions will pull electrons in a specific direction to allow that activation energy to lower even more. So things like B vitamins basically allow the fit of the substrate much easier, and you'll see that on this animation. So you got your cofactor. I'm going to show you a little video. This is actually in your book, and on your online component book, but I'll show you right now.
This animation demonstrates how B vitamin coenzymes must be present in order to enable some enzymes to carry out their functions. Many of the B vitamins assist in the complex biochemical processes that produce energy from food. The B vitamins are essential parts of coenzymes. Coenzymes are part of the machinery that allows reactions to take place. Enzymes are large protein molecules that catalyze biochemical reactions by lowering activation energies. Enzymes accomplish catalysis by configuring chemical reactants in the best possible spatial arrangement for reactivity. This enzyme catalyzes the release of carbon dioxide when producing energy from carbohydrates. Combining with the coenzyme helps the inactive enzyme achieve the appropriate three-dimensional configuration. For this reaction, thiamine must be present as the coenzyme. The green coenzyme molecule above is thiamine pyrophosphate, the cofactor needed to permit the enzyme and the substrate, pyruvate, to combine in the correct orientation to catalyze the reaction. Now all three molecules are nested together, and the substrate, pyruvate, combines chemically with the coenzyme, thiamine.
I told you that this whole idea that there's an enzyme, a substrate, and an active site? And so like it was labeled down there, it's called the enzyme substrate complex. If you ever get referred to as known as the enzyme substrate complex, it's the idea that the substrate itself is actually connected to the active site and it's going through the process of the breakdown of the substrate.
A chemical reaction can take place and energy as ATP and CO2 are produced as products of the reaction. Once this happens, the remaining complex, containing the two-carbon fragment, thiamine, and the apoenzyme, rearranges and releases the two-carbon fragment, acetaldehyde, and the ATP. The departure of the acetaldehyde opened the thiamine pyrophosphate to combine with another molecule of pyruvate, and the cycle continues. Meanwhile, the energy is now in the molecule ATP and is available to do work or to be stored. Another way that a coenzyme can influence the reaction between substrate and enzyme is to cause a change in configuration of the enzyme without interacting with the substrate.
Come back to it. So that is going into the next set of environmental factors. I'll show you the rest of the video. The first one's known as an inhibitor. And I kind of showed you all a little bit this idea here. But basically, there's two different types of inhibitors. There's a competitive and a non-competitive inhibitor. Hey, fun noodles. Okay, so again, remember this is for a protein. I'll show you this idea that like denaturing allows the protein to basically break down and it breaks apart. Right? So if this is your protein, this is your enzyme. What's this part right here called? The active site. Good. So this is going to fit here. So obviously this is what? The substrate. So the substrate's going to come in here. It's going to break apart. It's going to whatever that enzyme is supposed to do. Okay. So now there's two different types. There's one that's called a competitive inhibitor, and a competitive inhibitor, what it does is just sits into the active site area. The substrate itself cannot then fit into the active site. Does that make sense? So that's competitive inhibition. It's actually sitting in the active site itself because it's competing for the same space. Okay. So non-competitive then does what? Does it sit in the active site anymore? No. What it's going to do is actually it's going to change the configuration. And it's almost always, and just like how it shows up here, what happens is the inhibitor can go into what's called a non-competitive inhibiting. So it sits itself right here on the very top of it. It changes the shape of that enzyme, and again, the substrate will not be able to fit into the enzyme. So a non-competitive inhibitor sits somewhere else on the enzyme. It actually attaches to the enzyme itself, but does not sit within the active site. Yes? It's kind of like at lunch table where you're like, hey, this is my seat, and you go and you get some more food and you come back. If someone's actually sitting in your seat, what kind of inhibitor is that? Competitive. But if someone comes in and you go up and you go grab your food and you come back, and everything kind of shifted to the right, and so your seat no longer kind of fits in anymore because someone else sat down but they didn't take your seat, but your seat's still there, but it's kind of kicking out now, and so no longer do you have that nice little fun little round table, and now you have a round table with a little seat kind of kicking out a little bit. Okay? You're no longer sitting in the table, right? You no longer get to hang out with your friends anymore because what you have is now what kind of inhibitor? Non-competitive. Because you got your seat, awesome. Hey, I'm going to sit over here, guys. Hey, what's going on? Okay? But it's because someone else came in and brought their own seat into it, and it's changed the structure of that table. Okay? And also what this picture is showing as well is what's known as allosteric inhibitors. Okay, an allosteric regulator. What an allosteric regulator does, it actually changes the shape of the active site. Okay? So allosteric inhibitors, it can also be an allosteric promoter. What happens is that proteins themselves, again, remember proteins are just folding and bending and alpha helixes and beta sheets and all these different things, right? And so what happens is that you can have an inactive form of an enzyme, and you can have an active form of an enzyme. And here in just a second, we're going to look at what's called cooperativity and feedback and inhibition. And what can happen is that you have an enzyme. There's an enzyme above that. I'll show you the picture. Okay, so you have an enzyme. That enzyme goes into initial substrate. Until that initial substrate breaks into a new substrate, this enzyme will not work. Okay, it has to actually change its shape because of allosteric regulators to then activate this enzyme. Okay? So you can either use an allosteric regulator to inhibit the production, the use of an enzyme, or you can use an allosteric regulator to allow for the production with the use of an enzyme. Okay, so it actually changes the true structure of the enzyme. Okay, so this is what's known as a feedback. This is a positive feedback. Initial substrate comes in, makes another substrate, and it ends up with a final product. This happens a lot. Okay, and again, remember I talked about what's called there's ATP, and there's ADP, and there's cyclic AMP. Cyclic AMP is actually a messenger, and that cyclic AMP would be one of these first substrates, and it would actually fit into one of these enzymes. Okay. But what can happen is this is a positive feedback. This is a negative feedback. So again, we're making this end product. Okay, so it makes more, it makes more, it makes more, and so eventually this needs to shut down. This is like if you are breaking down glycogen to produce glucose. Okay, well eventually what happens? You're going to have too much glucose. And your body's like, wait, we have too much glucose, we need to stop. What happens is that if it makes too much glucose, it can then sit back here on this first enzyme. It changes the shape of the enzyme, and that initial substrate never actually goes in. So all of a sudden now you get put the brakes on. This is called negative feedback. Okay, there's both positive and negative feedback. So it changes, this final end product will actually change the shape of the first enzyme and allow it to stop. Okay. Whereas, and I kind of skipped on cooperativity, but it's the same kind of idea as the coenzymes. Cooperativity basically just says that there's some kind of extra part within the active site, on the outside of the active site, both ways, and it allows for that substrate to occur. I'll show you the rest of this.
This animation demonstrates how B vitamin coenzymes must be present in order to enable some enzyme. Another way that a coenzyme can influence the reaction between substrate and enzyme is to cause a change in configuration of the enzyme without interacting with the substrate. Here you see the conformational change in the enzyme needed to configure the enzyme to accept the substrate in a lock and key complex. This is called an allosteric control. The cofactor approaches the inactive enzyme and binds to it in an enzyme-coenzyme complex. The new configuration is a good fit for the substrate. Once this enzyme cofactor substrate complex is formed, energy can be released as ATP, and the other reaction products can be released from the complex. Now the correctly configured active site on the enzyme is free to engage another substrate. The amount of B vitamins that are needed to serve as coenzymes is quite small. There is enough thiamine activity in 1.2 milligrams to meet the average person's needs all day. Extra B vitamins will not produce more energy. Excess B vitamins in the body will pass out of the kidneys into the urine. This table shows some details about several of the roles played.
So what does that tell you about those energy drinks? They're like, oh, they're packed with like B12 and B6, and oh, it's going to give you... Does it really help you? Yeah, it just makes you pee it out. Exactly. Okay. A, D, E, and K, vitamins A, D, E, and K are actually fat-soluble vitamins. Those ones you don't pee out. Everything else, if you have too much of it, it just flushes out. Like vitamin C, I'm going to take lots of vitamin C and I'm going to stay healthy. Not really. This is going to make your pee darker. That's all it's going to do. So you're basically buying vitamins, right? And I'm not saying that your multivitamins are not good, because there are extra things in there. You know, those ones up there that are there to basically, if you don't need a complete diet and those kind of things, that they can help you. But a lot of times, especially if you buy like lots of vitamin C or whatever, it does, a lot of it is basically just making really expensive urine. That's all it is.
By B vitamins as coenzymes.
Go away.
So do inhibitors ever link the enzyme and like to get back to how it was before?
It can, yeah, and that would be then a negative feedback. And so basically that inhibitor could be like I was talking about right there. And so if it is a negative feedback loop, the inhibitor would be the negative feedback component. So remember on that slide, can't get out of this now. So like right here, this right here would be an inhibitor that's stuck onto here. Okay, and so what happens is that, let's say again, this is glucose that's being released. Okay, so we've released enough glucose, and now we've slowed it down. All of a sudden now your body needs more glucose. There will be another component that will actually pull that piece out, and then allow for the enzyme to continue on. So there's both regulators to stop it as well as make it then go on. Okay. All right, any other questions?